I'm just going to introduce our project in which we've generated human sensory neurons from induced pluripotent stem cells in order to generate a myelinating co-culture system. And the reason we wanted to do that is it's really essential for healthy peripheral nerve function, a very close and intimate relationship between axons and Schwann cells. So axons are myelinated by Schwann cells, and there's complex bidirectional signaling between these two different cell types. Now that signaling can go wrong in disease states such as inherited neuropathy, inflammatory neuropathies, and we obviously want to understand that better, preferably using a humanized system in which we have the native proteins in their normal environment. And so to do that, we've taken advantage of really this emerging technology, which is induced pluripotent stem cells. The principle there is we will take a skin biopsy from a patient, generate fibroblasts, and then we can reprogram those fibroblasts with a number of oncogenic factors. We can then generate pluripotent stem cells that can give rise to any cell type in the body, but in this case we've differentiated them into sensory neurons. And then the aim of this project is to combine these sensory neurons with rodent Schwann cells and really to understand if we can generate long-term myelinating cultures using this system, and most importantly to see if we can use it as a model system to study disease.
In order to establish the myelinating co-cultures, we first differentiate the iPS cells to sensory neurons, mature them for several weeks, and then seed in the primary Schwann cells. The subsequent alignment and onset of myelination recapitulate exactly those same events that occur in vivo. So here you're seeing a live myelinating co-culture. You can see the individual human cell bodies, and the phase-bright elongated structures are the myelinated axons. We've performed cross-sectional electron microscopy imaging through these myelin segments, and you can clearly see the individual myelin wraps and the surrounding basal lamina. Between each myelin internode, we see successful formation of the node of Ranvier with correct compartmentalization of key nodal proteins, including voltage-gated sodium channels at the node and contactin-associated protein in the adjacent paranode. The Schwann cell and axon are in very close contact and are constantly signaling to each other. One crucial pathway vital in the initiation and maintenance of myelination is the neuregulin B receptor pathway. We've overexpressed neuregulin-1 type 3, specifically in the sensory neurons using AAVs, and we see a dose-dependent increase in the levels of myelin production. So this confirms that this vital pathway is active in our myelinating co-cultures. So we've shown that we can successfully model the interactions between a human sensory neuron and a Schwann cell in vitro. So the next step is to investigate a pathological scenario that impacts on the functioning of these two cell types.
The development of a culture system involving the myelination of human sensory neurons provided us with an invaluable opportunity to investigate the pathology of immune-mediated demyelinating neuropathies. In this study, we primarily focus on the chronic condition chronic ataxic neuropathy with ophthalmoplegia, M-protein, cold agglutinins and anti-disilocyl antibodies, also known as Canamad. The major reason for this focus is that this neuropathy has prominent and often disabling sensory dysfunction, as Tony Morris, who began to experience symptoms of this disorder in the early 1980s, illustrates.
In the early 80s, I started to feel my feet were very uncomfortable, and I was generally walking very clumsily. A few years later, the numbness and the lack of control moved up my legs. This meant I didn't really have any balance, so I couldn't stand or walk. And whilst initially things were very uncomfortable and there was a lot of pain and discomfort, once the peripheral neuropathy had spread throughout my entire body, I lost all sensation of pain. The whole of my body had lost its sensation.
By applying anti-disilocyl human antibodies to our myelinating co-culture system, we're able to demonstrate both the topographical targets of these antibodies in culture and also their pathological effects. Antibodies were found to target the exposed nodal and unmyelinated axolemma, as shown here. With the addition of a source of complement, acute and extensive axonal degeneration ensued. However, Canamad has demyelinating as well as axonal features. We speculated that if demyelination was also mediated by anti-disilocyl antibodies, then this may be via a more chronic, complement-independent mechanism. The impressive longevity of our cultures allowed us to continuously apply disilocyl antibodies over 2 4-week periods, both at the onset of myelination and also in established 6 to 9 month old cultures. These experiments revealed that this exposure both prevented myelination and also induced demyelination without affecting axonal integrity. This work has therefore established that anti-disilocyle antibodies have pleiotropic pathological effects. Co-cultures now provide an ideal system in which to further establish the mechanism of these pathological effects and also to test new therapeutic strategies.
In the early 80s, I started to feel my feet were very uncomfortable. When I had shoes on, it felt as if I had pebbles in the shoe. My toes were sore, and I was generally walking very clumsily. The following year, the similar symptoms in the hands, the fingers became sore, and gradually the fingers became numb, and then the hands became numb, and I didn't feel any joint sensation, I didn't feel any pressure on the feet, and it didn't happen overnight. It was taking years to progress, but as a few years later, the numbness and the lack of control moved up my legs. It could be tracked because I lost the hair on the legs. Similarly, I lost all the hair progressively up my arms. If you can't feel your hands, I could see them and they would do what I wanted them to do, but if I couldn't see them, I didn't know what they were doing. This meant I didn't really have any balance, so I couldn't stand or walk unless I was holding on to something. And in order to walk, I had to see my feet. So I tended to wear white trainers because I could see them in my peripheral vision. If you can't feel the pressure on the joints, you don't know whether your foot's on the ground or not. And I had several falls because I tried to walk, but my foot was stuck to the ground because I was still standing on it. So I very quickly learned to walk with a very plodding gait, and with the white trainers I could see that my foot was lifting off the ground because I couldn't sense it myself. Obviously the clumsiness and lack of finesse in the hands made everything in life difficult. Holding a knife and forks, coordinating to get the food to my mouth, cleaning my teeth. I could only grasp the toothbrush with my fist, and I had to look in the mirror to find where my mouth was. So life was very clumsy with everything. I lost the sense of grip. I could grip if I could see what was there, but I didn't know whether I was crushing it or whether it was just going to slip out of my hands. And obviously, when it progressed, the hands lost all their tactile feel. They became very dry, and I lost any. As it progressed, the skin didn't perspire, so I started to have problems controlling my temperature. I was treated with various treatments. Some were very successful. Steroids, for instance, prednisolone. Initially, I really felt a big improvement and recovery of the peripheral nerves, but within several years, they were losing their effectiveness, and in the end, they had to look for alternatives. And after trying several other treatments, they eventually found immunoglobulin had a very beneficial effect. But again, after a number of years, that started to lose its effectiveness. So by this stage, we're now talking approximately 16 years, the whole of my body had lost its sensation. And whilst initially things were very uncomfortable and there was a lot of pain and discomfort, once the peripheral neuropathy had spread throughout my entire body, I lost all sensation of pain, which was a benefit because life was a real struggle. The only things that I was aware of that weren't affected was my hearing. My optic nerve still worked, so I could see, but I couldn't feel my eyes, and I couldn't, the eyelids drooped and the muscles controlling the eyes were a problem. But I could sneeze, but I couldn't cough. It was quite a shock when I sneezed and everything worked. I lost the ability to, my face didn't exist. I couldn't feel it. And amongst other things, I'd lost my reflexes and my blink sensation. So even to this date, I can touch my eye and it doesn't particularly trigger a blink response. With regard, it wasn't just the skin that was lost. I lost sensation in the muscle, all the joints. I wasn't aware of the position. The swallowing was very difficult, and I could only swallow liquids if they had been thickened. I lost the control of my tongue with regards being able to speak and manipulate food. A lot of problems with choking. I lost my sense of smell and taste. The reflux was a serious problem, and the whole function of my internal organs were being affected at the final stages. When you don't feel anything at all, it was as though it was my brain was being carried around by a robot, because I could see my body and I could tell it what to do, but if I couldn't see it, it would drift off, and I had no idea where my feet or arms were if I couldn't see them. I was then eventually diagnosed with a non-Hodgkin's lymphoma, and after a course of chemotherapy, I started to feel some recovery of some of the peripheral nerves. So life started to improve after that, and there has been a degree of recovery, but there are a lot of areas of my body that haven't recovered. So there's permanent damage, but I've got much better feeling in my hands. Similarly with the upper torso and my face have recovered to quite a good extent, but without the legs working properly, not having any sense of balance, it impairs everything. Everything is affected by the condition.